Okay, so dear students, so today I will be talking about another topic, which is microbial growth. So in the previous lectures, I talked about microbial nutrition. So there I talked about different points, like what is the different requirements, macronutrients, and how they are being taken up by the microbes, and what they are, how they are being metabolized. That we'll talk at some later point. So those kind of nutrients, they are taken up, they are utilized for the growth of the organism. So let me share some slides regarding this. Okay, so this is today's topic, microbial growth. So as all of you are aware that microorganisms, they take up different kind of nutrition that ultimately are metabolized, and that leads to the growth of the organism. So if we define growth, then what is growth? Growth will be defined as an increase in cellular constituents. By this I mean that the cell takes up different kind of nutrients metabolized, and that will lead to the increase in the overall size of the organism. It could be in terms of the cellular organelles, in terms of the overall size. So this growth could lead to increase in the cell number as well when the microorganisms are adult enough and they reproduce. So this growth can be of two types. Either it could lead to the enlargement of the cell, when the cell is longer or bigger in the size, or it could lead to the increase in the cell numbers. So now there is another term which is called synocytic. So it's in multinucleate organisms in which chromosal replication is not accompanied by cell division. So there, the growth results in an increase in the cell size, but not in the cell number. So this is one typical example where the microorganism is taking up all the nutrients, metabolizing it, and leading to the growth in the size of the cells. So whenever there is a kind of need for the growth or the development of the cells, that it is usually not convenient to investigate the growth and reproduction of individual organisms. The reason is simple, that these are minuscule, they are very small in size, so it's really difficult to investigate the small microorganism. So what is usually preferred is that the whole culture, or in a way, the whole bunch of cells are investigated. So now there's another term which is called cell cycle. So cell cycle, it is the complete sequence of events extending from the formation of a new cell through the next event. So it means that once a single cell has been divided into two cells. So now the daughter cells are formed. From those daughter cells, the cell cycle starts. So it will ultimately pass through the maturation to the age, to the time point where it is fully matured. That will lead to the development of the cellular components, cell wall, cell membrane, and other organelles. And when fully matured, it is ready to divide further. So when it is ready to divide further, and it leads to the formation of two another cells. So that whole process, right from the formation to the next division, is called the cell cycle. So let's have a look at the bacterial cell cycle. So there are a couple of ways in the bacteria where the cell cycle takes place or cell division takes place. It could be because of the budding or fragmentation, but the most popular one is the binary fission. So what happens in binary fission is that one bacterial cell divides into two bacterial cells. So you can look at this particular image here. So this is a representative bacterial cell where we can see that outer is the cell wall, cell membrane, and this is what we have the DNA content or the chromosome. and the different ribosomes. So when the cell is young enough or it is mature enough at an early phase of cell cycle and it is ready to divide, then the division process is kind of facilitated by division of the nuclear content or the DNA of the bacterial cell. This division leads to the elongation of the cells also. So once they are elongated to a certain length, then this constriction starts, the formation of a septum. Now this septum ultimately divides the two bacterial cells. And also the DNA content, which was kind of under the division process, separates and moves into two different daughter cells. And ultimately, that septate leads to the formation of two separate daughter cells. So, although cell cycle has been investigated in a lot of organisms, but in some of the organisms, as E. coli, Bacillus subtilis, and the Colobacter crescentus, they have been extensively studied. So, when we talk about the cell cycle, now there are two major points that need to be taken care of. Number one is the replication and partition of a DNA into the progeny cells. So it's kind of two-step process. Although they are going hand in hand, they both start together. So one is the replication and partition of DNA, another is the cytokinesis. So it's the formation of septum and the progeny cells. So when we talk about the chromosome replication and partition, so all of you might be aware that the bacterial chromosomes, or the DNA content is the circular one. So in that circular chromosome, there is a single site at which the replication starts, which is called the origin of replication or simply the origin. So replication starts at this origin and completes at the opposite end, which is known as the termination, or which is opposite to the origin. So this kind of replication is facilitated by a group of proteins which facilitates the DNA synthesis. So these proteins, they assemble at the point of origin and they are known as repliosome. So DNA replication, it proceeds in the both the directions from the origin, and the parent DNA is thought to spool from the repliosomes. So this is a typical representation of the chromosome replication and partition. So you can see here that this is the circular chromosome or circular DNA which is inside the bacterial cell. So this is one point of origin, and opposite to this origin is the terminus. So when the division starts, this group of proteins, which is called as the repliosomes, they come and bind to this particular origin. So two origins are there, and there is initiation of replication. So these chromosomes, they starts to separate, and these are kind of doing synthesis in the opposite direction. The cell also enlarges, it elongates. So when the cell has reached to a certain threshold length, so it means that that particular threshold length is required for the initiation of septum formation. So here, this is kind of this chromosome division or chromosome replication is already going on, and when after reaching a certain length, now the formation of septum also starts. So this septum formation is also kind of leads to formation of two cells where the chromosomes are already separated. And once this fully constricted, the septum, it leads to formation of two new daughter cells. So this is how the replication is going on. So now, after the formation of these chromosomes, now this second process, which is the cytokinesis. Although I told you that the chromosome replication and the cell division are going hand in hand, but we will talk about it at different times. So in this cytokinesis, there is important process which is called the septation. So it is the process of formation of a cross wall between the two daughter cells. So this septation is divided into multiple steps. So the first one is the selection of the site where the septum will be found. So it is not that it will kind of start forming at any point. So there are specific mechanisms, there is a specific way which selects the site where the septum will be formed. So second is the assembly of a specialized structure called the Z-ring, which divide a cell into two by constriction. So there is a Z-ring formation that is taking place after once the site is decided. So these particular Z-ring formation at that particular site ultimately leads to the restriction of constriction of that particular bacterial cell. So third step is the linkage of the Z-ring to the plasma membrane and perhaps the components of the cell wall. So this particular Z-ring, this is inside the bacterial cell. So now it attaches to the plasma membrane. Because ultimately constriction has to be taken place from the plasma membrane followed by and also the cell wall. Now this particular step is followed by the assembly of cell wall synthesizing machine. So that cell wall synthesizing machinery is kind of responsible for the formation of cell wall, cell peptidoglycan, and also the other constituents. And the final step is the constriction of the cell in the septum formation. So ultimately, this Z-ring, which was attached to this bacterial cell, leads to the complete constriction and formation of two new daughter cells. So this is a typical representation of bacterial cells. Initially, it was thought that the bacterial cells, they don't have any kind of cytoskeleton. So if you recall, the mammalian cells or the eukaryotic cells, they have two different kind of proteins, two different kind of elements which controls the cytoskeleton. One is actin, another one is tubulin. So similar to those actin and tubulin, it was observed that these bacterial cells, they also have some kind of homologs which are similar to actin and tubulin. These homologs, they are named differently and they control the overall structure of the cells. Why? Because these kind of bacterial cells, either they are rod shaped, spherical or other shapes, and they have their constant shape. They have a specific kind of architecture. So a protein or some kind of element is required which could retain the shape of the cells. So if you look at this particular image, first one, here we have this MREB, which is kind of homolog to the actin. So it forms a spiral-like filament. And this spiral-like filament, it is kind of responsible for maintaining the overall shape of the structure. And this also serves as a pathway to facilitate the movement of chromosomes to the opposite poles. So initially the development may start some point here. Chromosomes, when they are matured, when they are developed into two, their application has taken place. When they are into two, then this facilitates the movement to the opposite poles. Now in some other bacteria, there is another one which is called the tubulin-like protein, which is called FTSZ. So this FTSZ assembles in the center of the cells. This FTSZ forms the Z-ring. This purple kind of thing, it is the Z-ring. So this particular Z-ring is responsible for the septation formation. Now there's another protein which is called the MinCD. So this MinCD along with another MinCD proteins, they oscillated from one pole to another pole. So this oscillation of MinCD protein kind of facilitates the binding of the FTSZ at the center of the bacterial cell. So in a way, they are kind of spring-like structure, which is a helical or coil-like structure, which is preventing the movement of zinc to the poles. It kind of restricts it to the center of the bacterial cell. So now this cell division apparatus, it has a lot of proteins which are involved. So this apparatus is composed of numerous proteins that are thought to assemble in the order shown. So if you look at this, there is a FTZ ring formation, which is followed by the binding of non-other proteins. This process of ring formation begins with the polymerization of FTZZ to form ring. So then there is a sequence of proteins like FTSA, ZipA, FTS. So these different kind of proteins, they anchor to this FTSZ ring. This anchoring facilitates the formation of the septum and the overall synthesis of the cell membrane as well as the cell wall. So there are some of the functions are that anchoring Z ring to the plasma membrane. These protein functions, they coordinate septation with the chromosome segregation. They lead to the formation of the peptidoglycan, and also they hydrolyze the peptidoglycan to separate the daughter cell. So it's not only that they are leading to the formation of plasma membrane and the cell wall, they also leads to the hydrolysis, which could lead to the separation of the two daughter bacterial cells. Okay, so now we talk about the growth curve. So bacterial populations usually studied by analyzing the growth curve of the microbial culture. So as I already told you, that these organisms, they are not studied as individual entities. They are studied either in growth or you can say in batches. So when microorganisms, they are cultivated in liquid medium, they usually are grown in a batch culture. That is, they are incubated in a closed culture vessel with a single batch of medium. So this statement I need to say is that if somebody wants to culture the bacteria, so what they do is they prepare fresh media. They can take them to the culture tubes or flask, whatever the requirement is, and they inoculate or they add a single batch of bacteria, or you can say a single colony to that particular batch of medium. So that we call as the batch culture. So this is a typical growth curve. So if you look at this particular growth curve, so here we have the log number of viable cells, and here we have the time. So if we draw a curve between time and the log number of viable cells, then we observe 4 different phases. So number 1 is the lag phase. So lag phase, as you can see, this is kind of a horizontal line where there is no increase in the number of cells. Then it is the exponential phase or the log phase. So in this particular phase, there is rapid development or rapid increase in the number of cells. So this particular increase is kind of constant with respect to the time. And after a certain time point, there comes the stationary phase. So again, the stationary phase is almost similar to what we have in the lag phase. That is, there is no increase in the number of cells. So after the stationary phase comes the final or the 4th phase of the growth curve, which is called the death phase. So in death phase, the total number of bacterial cells from here goes down. So with respect to time, you can see that these 4 different phases, they are usually followed when the culture is being done in the closed vessels under those conditions. So let's see these kind of phases one by one. So the first one is the lag phase. So lag phase is when microorganisms are introduced into fresh culture media. So when they are introduced in fresh culture media, there is no immediate increase in numbers. So what could be the reasons? The reasons could be that cells, they have been stored, let's say, for a prolonged duration. They may be stored at, let's say, some glycerol stocks, or they are available as powder form. So what does it mean? That when they are stored, they may be old. They may be old, they are depleted of ATP. Also, they may be lacking some essential cofactors and ribosomes. So before the cells starts to divide, these must be synthesized. So most of the time, the media contains different kind of constituents, but still, these cofactors and ribosomes, they need to be synthesized for the cells to divide, for their numbers to increase. So the media may be different from the one the microorganisms they were growing in previously. So what I mean to say is that either they were in those first stops or they may be in some other media. So they needs to be grown in a different media. Let's say we have provided a media which is differential or which is kind of nutrient rich. So it may take some more time for those bacteria to adjust. So when they are moved to this particular new media, then new enzymes they would be needed to be synthesized. So as you are aware that microorganisms they are usually growing in nutrient poor conditions. So now if you are providing them nutrient conditions where nutrients are good enough or are available in abundant, they need to synthesize a different set of enzymes. Or it is possible that the microorganisms have been injured and require time to recover. So if you are transferring microorganisms from one point to another, transporting them, so it is quite possible that they might be injured. So these are the 4 different points which could lead to the lag phase or there is no increase in the number of microorganisms. So second one is the exponential phase or the log phase. So in this, the microorganisms they are growing and dividing at a maximal rate possible. So this maximum rate depends on the genetic potential of the microorganisms, the nature of the medium, and the environmental condition. So if the media is kind of nutrient content rich, then the microorganisms will grow. And also the environmental condition is the temperature, and that is also available. So the rate of growth is constant during the exponential phase. So they are completing the cell cycle and doubling in number at regular interval. So let's say, for example, the bacteria E. coli, it has a doubling time of 20 minutes. So every 20 minutes, it is kind of dividing. So let's say if you start with 2 cells, so 2 becomes 4, 4 becomes 8, 8 becomes 16. So that after every 20 minutes, this particular doubling is in there. So the population is most uniform in terms of chemical and physiological properties. So what does it mean is that since the availability of nutrients and the environmental conditions are optimum, the population which is dividing at this particular point is uniform. So it means that there is no mutation or there is no variation in the daughter cells from the parent cells. Exponential or the logarithmic growth is a balanced growth. So it means that all the cellular components, they are manufactured at constant rate relative to each other. So if you look at the cellular compartments or if you look at the cellular constituents of the bacterial cells which are being synthesized in this phase, they will be same. So if the nutrient level or the environmental conditions change, then the unbalanced growth results. So let's say this temperature is being changed after a few minutes. So it will lead to the unbalanced growth. So now this unbalanced growth is usually observed in 2 types of experiments. So what are those? The number one is the shift up. So when a culture is transferred from a nutritionally poor medium to a richer one. So let's say most of the time the bacteria or the microbes, they are growing in nutrient deficient places. So now from there, if they are transferred into nutrition poor, it means there will be shift up. So they will start taking those nutrients, they'll keep on dividing. So there's another thing which is called the shift down. So when a culture is transformed from rich medium to a poor. So let's say a medium A is prescribed for a microorganism. So now from medium A, you transfer that particular microorganism to a nutrient B, which is kind of nutrient deficient for that particular bacteria. So if that is bacteria, then there will be shift down. So once the cells are able to grow again, balanced growth is resumed and the culture enters the exponential phase. So in both of these conditions, shift up and the shift down, so the bacteria has to do some extra work. So what the extra work is, that it needs to synthesize some other enzymes which could deal or which could tackle those kind of situations. So when there is tackling or when there is kind of those enzymes or those proteins are synthesized, then ultimately the growth, normal growth will resume. So at sufficiently high nutrient level, the transport systems they are saturated and the growth rate does not rise further with increasing nutrient content. What is meaning that the growth of bacterial cell depends on the availability of the nutrients. However, when there is high availability of nutrients, so that will not lead to the maximal. After there is a maximum growth, so after that there will be saturation in growth. So let's say even you might have noticed that whenever, let's say, we have a cow or a cow at home or a buffalo at home, so if we feed that particular cow, so initially it eats a lot. The speed of taking food is quite high, but after a few minutes, the speed slows down. So even if the whole basket is full of food, but it will not be eating at that faster pace. So the similar kind of thing is happening with the microorganisms as well. So this is a particular nutrient concentration versus growth. So here you can see the number of cells or the yield of cells per milligram of an. So when this nutrition is, this concentration per milligram keeps on increasing, there is increasing number of the cells. However, it is kind of exponential growth. So if we talk about in terms of growth rate means that the number of cells which are dividing. So let's say in 1 minute we are getting that rate is high. Initially it is high, and then it kind of saturates. So you can see that initially it was the log phase, and then comes the stationary phase. So even if this availability of nutrients is increasing, their growth rate is not increasing. So from 10 it becomes 20, from 20 it becomes 40, from 40 it becomes 80. So the growth is kind of constant at that particular. It is not as such they are increasing with the increasing concentration. Now let's say initially we have 10 microorganisms and the availability of nutrient was less. So if we increase, so it is quite possible that not all microorganisms they are dividing at the same place. They may be needing some more time to divide. However, when the nutrient condition reaches to the optimum, let's say at this particular point, so they may start dividing. Even you can see this difference between the points. Okay, so the next one is the stationary phase. So in a closed system, the population growth eventually ceases and the growth curve becomes horizontal. So this is usually attained when the bacterial population is at the number of around 10 raised to power 9 cells per minute. So what does it mean? That the total number of wild microorganisms remains constant. Now this is constant. Why it becomes constant? So it is quite possible that there is a balance has been established between cell division and cell death. Or in a way we can say the population has simply ceased to divide. They become metabolically active. So there are a number of instances where it could be there. So let's say the availability of nutrient is now limited. So initially there were less bacteria, more nutrient or more food was available. So they were taking up at a rapid pace and they were dividing. But now the bacteria are more and food is less. So the division has ceased. Also, the bacteria which were dividing, they were releasing toxins. Toxins were released, and those toxic substances now they are inhibiting the growth of the bacteria. So in a way, we can say that either the death of the bacteria and the cell division they are at equal rate. So initially it was the division was or the cell division was more, death was less, but now it is almost balanced. So bacteria in a batch culture may enter stationary phase in response to starvation. So when there is deficient of nutrients, then it will lead to the starvation. So starving bacteria, they produce a variety of starvation proteins, which make the cells much more resistant to damage. So it means that these particular bacteria, when they are at stress, it could lead to different kind of formation. For example, they can lead to the dormancy. So that dormancy will lead to the formation of spores. And those dormant spores, they are very hard to kill. They are very resistant to different kind of stresses. They can easily bear temperature, humidity, those kind of stresses. So now we have something called the DPS. DNA binding protein from start cells. So these DNA binding proteins or DPGs, they bind to the DNA which is responsible for protection of the DNA from any damage. So you can look at this particular figure here. So either we can have two, three different things which could happen when the cells are starved or this kind of growth arrest. So number one is the death phase. So in this, some of the microorganisms they will die and some of the organisms they will be survive. So either they have adapted to those toxic metabolites which were present or they have reduced their metabolic activity so now they can survive. Second one is the genetically programmed sterility. So these organisms, they are kind of genetically programmed. So in a way, they can easily resist the stress condition or the less availability of the nutrients. Another could be that they will genetically program for the programmed cell death. So what does it mean? That they are kind of dying at the program method or in a sequential manner. In the end, and those cells which were surviving, they could consume those cells which are dying. So those cells which are dying, they could serve as the source of nutrient for the bacteria which were survived. So here in this particular, you can see that there is a prolonged decline in the number of cells. So instead of a distinct death phase, successive waves of genetically distinct population microbes better able to use the released nutrients and accumulate toxins survive. So if you look at this particular graph here, this is time versus overall. So if you look at the particular graph, this is I believe we can get multiple kind of graphs. So overall population size is kind of remains a little bit constant. Why this is happening? That let's say at this particular point there are some bacteria which are dying. So now those bacteria could be utilized by the bacteria which are surviving. So in a way, so those bacteria, as I showed you in the last slide, that those bacteria are capable of utilizing the bacteria which are dying, their waste products or their toxic chemicals they can easily be utilized by the microbes. So this is all which I wanted to share with you. So, so thank you very much for your time.